we're not sure, but it's actually great that now we finally get connected. I'm sure we'll meet each other, and probably many of you in the audience in the future meetings when this thing is over. But it's, after all, a very nice way to be able to share our research work with people from all over the world in such a special way. So I'm quite looking forward to it. Today, I will talk about one of the projects in my lab. It's about our efforts to try to tackle the so-called variants to function challenge for a gene called LIPA, which is a risk locus for human coronary artery disease. As many of you may know, the human genome-wide association studies have identified many disease-associated genetic variations in the past few decades. And these discoveries have been very fruitful because it really make us to be able to understand deeper about the disease mechanism and discover the new drug targets. However, the question remains to be challenging because a lot of the genetic associations were not really fully understood. So we are playing our roles in those efforts by focusing on this LIPA gene. Before getting into this project, I would like to give a general overview of the research going on in my lab. Okay. I started my laboratory at Columbia back in the summer of 2018, so time flies. It's been about 2 years already. My lab is interested in the role of macrophage in cardiometabolic diseases with an emphasis on the use of functional genomic approach. Here is our lab webpage. Please feel free to go in there and see who we are and what we do. We also put a lot of resources like our protocol and data analysis workflow under the resource tab and make it available to the community. There are mainly 3 areas of research in my laboratory. One direction is the project I'm talking about today. And the other one is about using unbiased genome-wide CRISPR screen for novel discoveries, in which case we, for the first time, established a genome-wide CRISPR screen in primary macrophages for the discovery of novel regulators of macrophage efferocytosis, which is an important macrophage function to maintain homeostasis. And defective efferocytosis drives many diseases. I talked about this work recently in the virtual cardiometabolism seminar, so feel free to find the Twitter handle. There should be a link for the video recording. So I'm not gonna touch base about this project this time. And then another big part of the research in my laboratory actually started from my postdoctoral work when we used human iPS-derived macrophages for disease modeling and the functional interrogation of human-specific non-coding elements, including genetic variants, as well as long non-coding RNAs. I established and published that protocol back in 2015. After that, we used the protocol in a number of original research papers. We also published review articles on the use of human iPS-derived macrophages. We have the protocol on our webpage too, if anyone is interested in using iPS-derived macrophages as a tool. So please feel free to let me know if you may have any questions or if there's anything we can be of help. So now I will begin to focus on this project in which we try to tackle how LIPA, a risk locus for coronary artery disease, may regulate macrophage function in atherosclerosis. I would like to give the take-home message because in that case, when you follow along, like you kind of know where we stand and can focus more on why it matters and how we get there. So basically, LIPA has been reproducibly identified by multiple human genome-wide association studies as a risk locus for human coronary artery diseases. The next question is usually like what the directionality is. That is whether the risk locus leads to higher or lower LIPA expression. So in our case, by using a lot of functional genomic data, public available and generated by ourselves, we were able to confirm that the risk alleles associated with human coronary artery diseases leads to higher LIPA and in monocytes and macrophages in a cell type specific way. We're also able to have quite a lot of evidence to support that a SNP RS1320496, I will call it 496 from now on, is the potential causal variant that explained for the association between the risk allele and the LIPA expression. Of course, what matters is how the risk gene affects cell function and the disease itself. With the directionality we were able to confirm, we used the overexpression system to drive increased LIPA expression. We were able to find that the increased LIPA led to increased macrophage proliferation, which is characteristics of atherosclerosis progression. Ultimately, the question is how the increased LIPA affect atherosclerosis, because this will establish the mechanism of the genetic association. We are still working on it, so we don't have a conclusion yet, but we have some data. So I will show what we have and share our insights. By all means, we haven't really completely tackled this challenge. We're still working on it, but I still would like to share the data to let the field really know like what the challenge is and how we tackle it, what we have learned, and also get feedback from this community, because I really think the more we delve into it, the more we found it's closely related to the spirit of immunometabolism. All right, for those of you who are not in the field of coronary artery diseases, what is coronary artery diseases, also known as CAD? So CAD is the narrowing or blockage of coronary arteries that supply the blood to the heart. It's usually caused by atherosclerosis. Atherosclerosis is the build-up of lipid and inflammatory cell-enriched plaques within the blood vessel. It can happen anywhere, but of course, when it happens in the coronary artery, it's more detrimental because it can cause angina, ischemic heart disease, and if the plaque fall off, it leads to myocardial infarction. Coronary artery disease is an interplay between it's caused by interplay between genetic and environmental factors. In fact, the heritability is estimated to be between 40 to 60% based on family and twin study. This is also why it matters to understand the genetic underpinning of CAD. So genome-wide association study have been a very powerful tool for understanding the genetic underpinning of certain diseases. What is genome-wide association studies? So GWAS identify the association between the genetic variation and the trace, and in this case, the disease of CAD. So in GWAS, the participants with certain diseases and without certain diseases are recruited. And millions of SNPs are genotyped by using genome-wide SNP arrays. Once the genotype being figured out, then if a certain allele for a certain genetic variation occurs more frequently in the disease population, like showing here for the T allele, illustrated in this figure, we would say this genetic variation is associated with the disease. And then this genetic variant also mark the region in the genome that we call it the risk locus. Why this even matters, right? This is because like we have already learned that any drug targets with genetic evidence of disease association are twice as likely to lead to a successfully approved drugs. So this is why we really would like to understand the genetic basis and genetic evidence of human disease. In fact, this approach has been really very powerful because it already identified many known genes important in the CAD disease pathogenesis without prior knowledge. So the hope is that by studying the additional loci, now it's over 160 of them, we will be able to learn more about the disease mechanism and identify additional therapeutic targets for human CAD. In our case, we are interested in LIPA as a risk locus for CAD. LIPA has been reproducibly identified to be associated with CAD in the past decade. Very strong GWAS signals. What's interesting is there is also strong evidence showing that the risk locus is not associated with lipid trace. This is good to know because when we, the lipid level is a known risk factor for CAD, and when a genetic evidence suggests that a certain risk locus is CAD specific, not related to the lipid trace, it probably means that this is a very specific mechanism for CAD only without affecting CAD pathogenesis through altering the lipid level. So there's a very interesting mechanism behind. Now, many of you may already have the question, right? Because I mentioned that GWAS will identify the association between genetic variation. So how we know that when we see the genetic variation, we know it is the LIPA gene, not like other genes in the locus. Normally, this can be a very sophisticated question. In fact, many of the CAD loci, people still don't know what the causal genes are. And there are many bioinformatic tools and pipelines being developed to try to annotate the candidate genes from the genetic variation being identified by GWAS. In our case, however, it's relatively straightforward for LIPA. The field pretty much kind of believe that LIPA is a causal gene, mainly because the genetic variation is intronic in LIPA overlap with enhancer landscapes. And the most important piece of evidence is that the lead SNP for CAD is also the lead SNP for the LIPA eQTL. This is another new concept. What is eQTL? eQTL identify the association between genetic variation and expression of gene as a trace. So like GWAS, it identified the association between genetic variation and disease or trace, while the eQTL identified the association between genetic variation and expression. So when I say this lead SNP is for both CAD and eQTL, it basically means that the same genetic variation pointed to the CAD disease association is also the lead genetic variation that seem to affect the LIPA gene expression level. So that would strongly suggest that LIPA is the causal gene for this genetic variation. So what do we have known about LIPA? Like many of the GWAS loci, we know very little about their function. LIPA is actually fairly well studied because it is really very important. So LIPA encodes for a protein called lysosomal acid lipase. The field typically call it LAL, so you may feel it's more familiar, while the LIPA is the gene symbol. It is essential because it is the only known acidic lipase that hydrolyze cholesterol ester and triglyceride in the lysosome. We have published a number of review papers and original research papers to tackle the role of LIPA in macrophages in the past. So here is a schematic figure from one of the reviews I published before to summarize what LIPA does. So basically, human cells will take up lipid from outside, mostly by endocytosis of modified lipoprotein. There are other mechanisms like micropinocytosis, but I try to make it simple. So basically, when the lipoprotein is being engulfed, will be delivered and fused with the lysosome through the endosome and the lysosome fusion. And then within the lysosome acid lipase in the acidic environment would hydrolyze cholesterol ester and triglyceride, release free cholesterol and fatty acid. The free cholesterol can be effluxed to leave the cells, or re-esterified and stored as lipid droplets. And the fatty acid can fuel the fatty acid oxidation, and they can also be the substrate for lipid mediator synthesis. So you can imagine that it is the only known acidic lipase in the lysosome to hydrolyze the lipoprotein. And the loss of function would definitely be really bad. In fact, loss of function mutation cause Mendelian disorders, including Wolman disease and cholesterol ester storage disease, characterized by hyperlipidemia, liver failure, and premature CAD, most likely due to the secondary effects of the severe hyperlipidemia. However, as the CAD-associated risk alleles, there's no association with any of the lipid traits, as I mentioned in the previous slides. Therefore, the lipase CAD association must be through an independent mechanism from a liver-mediated cholesterol metabolism. This is the first idea we've had. For this type of the variant to function question, when we tackle the genetic discovery from human GWAS, there are usually those three questions to be asked. So first of all, how is lipid regulated by those GWAS CAD variants? And what are the causal cell types and the causal genetic variants that regulate lipid expression? And then once we figure out the directionality, how does the lipase will affect the cellular function in vitro? And how does the gene will regulate atherosclerosis in vivo? So we spend a lot of efforts to try to tackle all those questions together. I will first start from the first question. How is lipase regulated by the CAD variants, and what are the causal cell type and the causal variant? So first of all, it's good that there are actually multiple studies published in the field to try to address the directionality between the risk allele of the lipase genetic variants and the lipase mRNA activity in human monocytes. You'll see I'm showing 3 studies here, including the recently published study by our collaborators lab, which shows that consistently the risk allele of the genetic variant associated with human CAD are linked with higher lipase mRNA in all those 3 independent studies. And in the last one, it also tested the lipase activity in the cell lysate. It's also consistently increased in the risk allele at carriers monocyte. This is important because this means it's not just the regulation at the transcript level, but it's at the level of the activity. So higher risk, higher lipase activity in the monocytes. And is it all about monocytes, not other cell types? Actually, yes. So this is a very nice data set. It's probably too small to read, but the take-home message here is that the genetic variant only shows eQTL in monocytes, but no signal at all in T cells or neutrophils. So this genetic variation and this regulation for lipase expression is at least monocyte specific if you consider the whole blood, the cell type in the whole blood. We were interested in macrophages because for human CAD, one of the disease characteristics would be the increase in macrophage accumulation in the plaque. The macrophage can be derived from the monocyte infiltration and differentiation, and also by local proliferation. So indeed, by using an in vitro model, we were able to find that during monocyte to macrophage differentiation in 8 days, the lipase expression very remarkably increased. And previous data set also shows that the risk allele of the genetic variants associated with CAD are linked with higher lipase mRNA in the macrophages. The most importantly, this is our own biobank of human monocyte-derived macrophages with the genotype array data available, and we were able to confirm that the risk allele associated with higher lipase mRNA and activity in monocyte-derived macrophages. And as I mentioned, the human GTAX data has a large-scale eQTL study for different tissues across a human body. And particularly, the LIPA eQTL signal is the strongest in the whole blood. And we already found that the signal must be driven by monocyte because there was no eQTL in T-cell neutrophil. And the signal is also very strong in the spleen. Spleen is actually full of macrophage. So if we look at the lipase staining in the spleen, we can actually tell that the lipase is abundantly expressed in the red pulp macrophages. There's no like cold staining, but just by looking at the pattern, it can be confirmed that it's about the macrophage lipase. Macrophage is also quite abundant in the lung, and as we can see, the lipase is abundantly stained in the lung tissue, and this is consistent with the GTAX showing the lung and the spleen as the organs with the most abundant expression of lipase. So the point of this data is we confirmed eQTL and activity association in monocyte and macrophages. And here with this GTAX data, we were able to get the clue that the tissue eQTL for lipase must be macrophage-driven. So now we have a clear clue about the directionality, risk allele, higher lipase. Next, we want to tackle what might be the potential causal variant that affect the lipase expression. You would probably think it could be straightforward because we know the lead SNP. Why not it just the lead SNP that regulate the lipase expression? In fact, it is not that straightforward. For example, I'm showing the lipase locus here. There are, in fact, a total of 13 SNPs in high linkage disequilibrium, with the GWAS lead SNP show the most significant association. What does this mean by saying linkage disequilibrium? It means that those 3 SNPs are evolutionarily inherited together. So if you know the genotype of one particular SNP, you should be able to impute to determine the genotype of other SNP. When performing all those genetic association studies, including GWAS and eQTL, the signal, in other words, the significance level of those SNPs in LD would be somewhat similar because it's all based on the association for their genotype. In that case, we actually cannot tell within all those 3 SNPs which one are the causal one that indeed affected the lipase expression. So this is a question we try to tackle as the next step. How people usually do it is by plotting the landscape of the enhancer region, enhancer mark, and the transcription factor banding. So PO1 and CBP beta are the key transcription factor essential for macrophage maturation and myeloid cells identity. So that was why we plotted all this data with overlap of the SNPs in LD with the GWAS lead SNP. So we prioritized 2 regions. This is somewhat subjective because we basically just try to find, for example, for this region, there are clear peak of PO1 banding. There are enhancer marks, and this region, not as dramatically strong peak for transcription factor banding, but enhancer mark. So we simply first started from those 2 regions and tested, well, they show the enhancer mark and regulatory mark, but are they really representing enhancer? So if you remember the 444 region and 833 region, and the fact that the 444 region has a stronger peak. And when we are doing the luciferase activity, try to confirm, and indeed we don't see enhancer activity in both regions in HEC cells, which is not really myeloid relevant. And it's nice to not to actually see any signal, but in the THP1 monocytic cell line, we are able to confirm that only the 444 region with the stronger peak show the enhancer activity. Now we focus on that particular region. When we further delve into that, we don't really want to miss any signal, and we notice another SNP, the 1320496. So if we look at the GWAS data and then the whole blood LIPA eQTL data, you will see that that particular SNP by the color code is not in high LD with other GWAS SNPs that cluster together. However, it did show an equivalent significance level in terms of the GWAS association and eQTL association. So that was why we kind of included the SNP in our next step of interrogation. In fact, there is also another reason, because PO1 shows a very strong binding peak in that region, and this particular SNP, in fact, the risk allele created the PO1 binding site. And here the data simply plotted the significance level of the LIPA eQTL and the expression level of PO1 transcription factor expression. And clearly it pulled out the spleen and whole blood that shows the strongest LIPA eQTL and highest PO1 expression. This kind of suggests that the eQTL in those two tissues are driven by the PO1 transcription factor. It is functional genomic data. It only suggests association. It needs to be validated, but this gives us the confidence to focus more on this particular SNP and PO1 binding. Indeed, the allele-specific PO1 binding ChIP-seq data also support that the risk allele of the 496 SNP showed a stronger binding with PO1 compared with the non-risk allele. This is within the same subject, so it's nicely controlled. When you do see differential binding, it strongly suggests that the risk allele has stronger binding with the PO1, and therefore very likely explained for increased LIPA expression. To provide further evidence, we performed the site-directed mutagenesis. Although there can be, mathematically, there can be different kind of recombination. In reality, the human genotype would only show 3 types of haplotypes. The non-risk haplotype, which means the CCC allele for 3 SNPs in the region, the risk haplotype, which would be TTT or CTC. So we wouldn't see TTC or any other combination, very unlikely in a human being. And then we perform the luciferase assay. So it is clear that these are 3 independent experiments. I try to plot the data from the same experiment together by drawing a line to connect them. And then it shows that the TTT haplotype increased LIPA enhancer activity, shows that the enhancer activity is stronger with this TTT haplotype. And for the CTC haplotype, it's also increased compared with CCC, but not as high as shown in the TTT, perhaps suggesting that the 496 SNP and the other 2 SNP has additive effects. One goes deeper for the so-called conditional analysis, which means we conditioned to first to only analyze the subject with the non-risk allele for the 444 and 445 SNP. The presence of the risk allele for the SNP 496 showed a higher LIPA compared with the CC non-risk allele. And then if we condition to the risk allele carrier for the 496, which are showing here, and then the presence of risk allele for the other 2 SNPs show the higher LIPA. This is just providing further evidence that the 3 SNPs may have additive effects on driving the increased enhancer activity by the risk allele. So much about the granular detail about the causal genetic variant, but it's interesting to learn the knowledge. After all, for the summary of the first question, we were able to determine that CAD risk allele in LIPA are associated with higher LIPA mRNA and activity in monocytes and macrophages. LIPA is abundantly expressed in macrophages and is induced during monocyte to macrophage maturation. Monocytes and macrophages are most likely the causal cell type for the CAD LIPA association. And then the SNP 496 is the potential causal SNP. And the risk allele create a binding site for PO1 and increase the LIPA enhancer activity. These are important to know, but in the end, what's more important is what's the functional consequence of the genetic variation. So after figuring out the directionality, we would like to address how increased LIPA affect macrophage function. We used the lentivirus-mediated LIPA overexpression in THP1 monocyte-derived macrophages. This is because for human macrophages, due to the high SAM-HD1 expression, they are very refractory to lentivirus-mediated overexpression. So we got to start from monocytic cell line and then differentiate them to macrophage to study the effects of the LIPA overexpression. You will see that the overexpression is not that dramatic. Perhaps because the baseline LIPA level is already very high. But if you remember the eQTL data I showed earlier, this 30 to 40% of the increase is actually equivalent to what we've seen in the risk allele carriers compared with non-risk allele carriers. So we think at least it resembles what we see physiologically in human macrophages. So we decided to go further with this model. What could LIPA overexpression impact macrophage function, right? So based on the knowledge we learned, we try to come up with hypotheses. The first hypothesis is relatively straightforward. Maybe many of you are already thinking. It is that with the LIPA overexpression, perhaps the hydrolysis of lipid in the lysosome is accelerated. Therefore, we get more free cholesterol and fatty acid, and increased efflux, and increased reesterification and storage of lipid droplets. So basically, the LIPA overexpression accelerated loading. In reality, we actually didn't see it. So we tried to do live cell imaging so that we don't miss any time point. We tracked the DO labeled acetylated LDL uptake, showed no difference. And then in the end, we also stain for neutral lipids droplets and quantified the fluorescence intensity, and it's also not different. So this is probably right because there's no reason that overexpression LIPA will, for example, feed back to the cell surface receptor to express more scavenger receptor for increased uptake. So we thought, yeah, that's possible. And then we thought about another hypothesis. This is based on recent work showing that oxidized lipoprotein loading would accelerate free cholesterol accumulation in the lysosome in particular. And when the free cholesterol accumulates in the lysosome, it will impair the proton pump function of the lysosome membrane located on the lysosome membrane, therefore impair the lysosomal acidification. So the lysosome pH would increase and subsequently impair the activity of other acidic proteinase. We thought that was a reasonable hypothesis and we tested. But again, when we were trying to determine the lysosome pH and proteinase activity, the chloroquine, which is our positive control that absolutely inhibits lysosomal acidification, we do see the increased pH and we see impaired proteasome activity, but overexpression did not change much. So at this point, we thought that maybe because of lipid loading, would itself trigger lysosomal biogenesis, as our colleague, Dr. Baker-Ozani showed in his publication, multiple publication. So probably we should focus on the baseline level without any lipid loading, because if lipid loading can trigger LIPA expression, then we overexpress it. Will that overexpression effects be kind of masked by the lipid loading induced effects? So then we thought, all right, without lipid loading, maybe the lysosomal LIPA overexpression will still accelerate the influx of lysosomal, for example, fatty acid into the cell, but then feed to the fatty acid oxidation pathway or provide increased substrates for lipid-mediated synthesis, which has probably profound effects, and maybe provide more substrates for cell growth. We actually don't know, but we decided to simply look it up. So indeed, we used lab cell imaging and determined the percent of confluence of the cell culture dish. And we found that the overexpression cells grow a bit quicker and plateaued at a higher confluence level. And then we determined the cell count because the confluence level may also be attributable to the cell size instead of the cell number. And again, the cell count showed a higher number in the LIPA overexpression cells. And then during the log growth phase, the EDO incorporation, which marks the cells undergoing DNA synthesis, is higher with LIPA overexpression.
Andrew, I think you are frozen. At least that's what I'm experiencing. Let's wait a bit to reconnect. Actually, I was also kicked out earlier during the presentation, so it's apparently a busy day on the internet. I will check. So this is the risk of online seminars. Let's hold on for a while before Hanrui can reconnect again. It might help to disconnect the cameras, but it shouldn't affect the online seminar. So please hang on and we will see. Meanwhile, you can raise your questions via the chat box. So let's hope she comes back. Okay, Hanrui is joining again. My co-host, yes. Hi, sorry. Yes. Hey, Hanrui.
Hi, sorry about that. Yeah,
No
somehow
worries.
I got kicked out.
No worries.
Okay.
It froze and then you were kicked out. Sorry for that.
Right, yeah.
But good to have you back. So
Great.
please start sharing again.
Sounds good.
I think we only lost 1 or 2 visitors, so that's nice.
Okay.
Thank you
Yeah.
all. And
Thanks for being patient.
yeah, thank you.
Technical glitch.
I will mute myself again and then you can continue your presentation.
Sounds good.
Thank you.
Mm-hmm. I'm trying to share my screen, so just bear with me. Almost there. Almost there. Okay. Okay. All right. All right, here it is. Great. All right, hopefully it's good from now on. Yeah, I think talk through that. Basically just overexpression, increased proliferation. And then we couldn't think of a mechanism at that point. So we decided to do RNA sequencing. We found that consistent with the phenotype we observed, we do see the top enriched GO pathway is the cell proliferation. So which kind of confirmed with what we see and confirmed the validity of RNA sequencing. When it goes deeper into the pathway analysis, we found that somewhat surprisingly, the PI3 kinase AKT signaling pathway and pathways for inositophosphate synthesis are upregulated with LIPA overexpression, as both shown here and here in the representative gene set enrichment analysis plot. That triggered us to do a targeted lipidomics panel to see whether phosphatidylinositol levels were indeed increased. So we were indeed able to see that phospholipids were modestly increased with LIPA overexpression, and then phosphatidylinositol was the only phospholipid species that upregulated with LIPA overexpression. So at that point, like, we were actually not sure what could be the molecular mechanisms, and this is what we are trying to dig deeper into it. But for what we tried, we were able to find out that LIPA overexpression did not accelerate lipid loading or oxidized LDL-induced lysosomal dysfunction, but it did, without the lipid loading, increase the macrophage proliferation, which is an interesting phenotype. And we found it's probably associated with increased intracellular phosphatidylinositol and PI3 kinase signaling, because those signaling pathways are indeed known to be positive regulator for cell proliferation. We are going to try to establish the connection at the molecular level. So while continue to dig into the in vitro cell data, we recognize the importance to move everything in vivo, because that will address the pathophysiological role and establish the GBA association and connection with the mechanisms. So the last question we were trying to tackle was how does increased LIPA affect macrophage function and atherosclerosis in vivo? That was around the time I established my own laboratory. I pulled the trigger to generate this mouse model by ourselves. So it is a ROSA26 knock-in for LIPA overexpression. So it is a CAG promoter followed by LOXP flanked stop codon, and then followed by lipase cDNA ORF and EGFP, but separated by iris, so that the cells express LIPA would be GFP positive, but the GFP is not fused with LIPA, therefore shouldn't affect its trafficking. By cross it to LysMCre, we will be able to induce myeloid-specific overexpression. So we, at that point, were only able to do bone marrow transplantation, which determines the overexpression in the hematopoietic compartment with the LysMCre specifically for myeloid cells. The reconstitution was great. In the peritoneal macrophage, we were able to see like over 60% turn out to be GFP positive, meaning that they should have the overexpression of LIPA. But the reconstitution was relatively lower in bone marrow cells, which is consistent with the LysMCre model after all. And probably partly because of that, we do observe increased LIPA activity, but relatively modest in bone marrow derived macrophages, or more dramatic in the peritoneal macrophages. Overall, we would hope that we will be able to drive the overexpression further, but at least what we found is consistent with the effect size we were able to have for human macrophages from risk allele carriers versus non-risk allele carriers. So we think we are resembling the physiological status. what we see this with this model, so we were indeed able to confirm that the bone marrow macrophages from those lipase transgenic mice showed modestly increased proliferation marked by the EDO incorporation. With the current recombination, we don't see any change in the hematopoiesis, both at the stem cell level and peripheral blood monocytes and neutrophil. But we were able to find that the peritoneal macrophage and myeloid cell numbers were modestly increased with the lipase overexpression. So all the phenotypes are modest, but again, we were using bone marrow transplantation, so the recombination was modest, although still resembled the physiological effect size. And then we didn't really have any hyper challenging with this model. So we hope that we will be able to get back to the lab and then repeat the experiment. But it seems like overall, we see consistent macrophage proliferation phenotype in vitro and in vivo. So why this even matters, right? What does macrophage proliferation do? So in atherosclerosis, macrophage accumulation is a major characteristic. It can be from monocyte recruitment and then local proliferation or impaired egress. So increased macrophage proliferation is absolutely associated with disease progression, and the pharmacological inhibitors or genetic manipulation that delay or inhibit macrophage proliferation has been shown to be atherosprotective in experimental atherosclerosis repeatedly. So we think that could establish a link between the genetic variation in lipase and the disease etiology. As I mentioned, we don't really have the data yet. We are still working on it, but we do have some insights to offer. First of all, in atherosclerotic aorta, the lipase is in fact higher. So we do see increased lipase in the atherosclerotic aorta. This is both in published data set and in our data set in Apple knockout mice, showing that with the longer time of Western diet feeding, we see a variable but a trend of increased lipase. Now the question is that increased lipase due to increased plaque macrophage number because macrophage abundantly express lipase, or because increased macrophage lipase expression per macrophage? So perhaps both. With the advance of single cell technology, we were able to do single cell sequencing of the plaque cells. And then here it's basically showing these clusters of cells are macrophage-like, while the other clusters include vascular muscle cells, endothelial cells, and T cells. So basically, macrophage show the most abundant expression of lipase, but only a subpopulation of this macrophage are the most abundantly expressed lipase. So perhaps both increased macrophage number and the increased expression of lipase in certain population of macrophage all contribute to this increased trend of lipase expression in atherosclerosis we observed. And from a published paper, it also confirmed that when they isolate foamy macrophages and non-foamy macrophages from the plaque, it is the foamy macrophages that express much higher level of lipase compared with non-foamy macrophages. So lipase is highly expressed in plaque foamy macrophages. And in our human atherosclerotic plaque samples, we do see abundant lipase expression and mostly co-localized with macrophage marker when we stain the macrophage with CD68, and this is human plaque sample. So what's the point, and what I meant to say is that based on our data, at least atherosclerosis is not perhaps due to the absolute deficiency or lack of lipase. It's possible that in atherosclerotic plaque, there's lysosomal lipid accumulation. So despite the fact that there is lipase in the lysosome, because of the impaired lysosomal acidification, the lipase is not functioning. However, we do have a lot of lipase. And what will happen if we further overexpress lipase, right? Based on the genetic association directionality from functional genomic analysis. So we are very enthusiastic to further address this question. All right, summary 3 for the question 3. we were able to confirm that LIPA transgenic overexpression increased bone marrow macrophage proliferation and increased peritoneal macrophage number modestly. And then with the current data we were able to see that LIPA is increased in atherosclerotic plaque macrophages. What we'll see if we further overexpress LIPA will be the next question we try to address. The last summary and future direction, we were able to use a variety of functional genomic tools to establish the directionality and the causal variant between the CAD GWAS association and increased LIPA, specifically in monocyte and macrophages. And increased LIPA expression drives increased macrophage proliferation, both in vitro and in vivo. We used lipidomics and RNA-seq to find that increased phosphatidylinositol and PS3 signaling might be the mechanism, but we still need to further delve into the molecular mechanism. And then our current work is really to address how increased macrophage LIPA would affect atherosclerosis. All right, lastly, this is the Columbia Medical Center campus. My lab is right in this building. And then this project is spearheaded by a very outstanding postdoctoral fellow in my lab, Fang Li. She joined the lab late 2018 and really moved the project forward significantly, quite fearless and very action-oriented. She also recently received the American Heart Association postdoctoral fellowship. I'm looking forward to see the story being further unfolded with the efforts. And this is also teamwork. Pretty much everyone in my group has contributed to the project. I also would like to thank all the collaborators, both at Columbia and other institute and the funding. Thank you.
Thanks a lot, Hanrui. This was pretty cool. Thank you for sharing this very recent data. I really liked how you go from GWAS to a mouse model and to in vitro models. Mm-hmm. We often go the other way around, but this is certainly something to consider. Mm-hmm. Very nice. There is questions coming on in the chat box, and actually one is also related with something I was wondering while you were presenting. So you now looked at these LIPA overexpressing macrophages and they proliferate more. The question is whether it also affects cytokine secretion. And personally, I was also thinking, okay, how does it impact on the macrophage response if you try to skew them? Mm-hmm.
Right, this is indeed something we kind of tested a little bit. It seems like we see increased inflammatory cytokine chemokine expression with the LIPA overexpression. And then the GO pathway related to oxidative phosphorylation is downregulated. Seems like with overexpression, they are being driven to more like inflammatory-like. Yeah. But how this make the connection between the phosphatidylinositol is something we still try to think about it.
Right, right. Yeah, I was also thinking, because there is obviously this work from Ed Pierce showing that LAL provides the fatty acids for fatty acid oxidation, and that in the absence of this pathway, you have an impaired IL-4 and thus this M2 macrophage polarization. So did you look into that direction? Do you plan to do that? Could that be part of your observations?
You mean how the macrophage in response to M2 type of stimulation, right? Yeah, because I mean, right, I think that seminal work from Ed Pierce's group basically suggests that L4 increase LIPA, right, and make them become more like M2-like and anti-inflammatory. And in our case, we would think when we overexpress LIPA, they become more M2-like, anti-inflammatory, but we actually see the opposite. So yeah, this is just what we see. Possibly, I mean, it's like the cause and the consequence, right? Like we overexpress LIPA, but it doesn't necessarily mean like it will affect macrophage M2 stimulation, although M2 stimulation can affect LIPA. So it's like we are manipulating the downstream of the L4 stimulation.
Yeah. I was also struggling with this kind of things. Xante van Dierendonk also asked a question relating to adipose tissue macrophages, who are also very relying on lysosomal lipid metabolism.
Right.
So based on that, you would also expect to see changes there. But again, there maybe overexpressing the system is not as, I wouldn't say efficient, but wouldn't yield the largest effects compared to knocking it down. So what's your opinion on this?
But I mean, I think we observe this a lot. Like if a gene expressed at relatively lower level, then the overexpression is generally more efficient. And you'll see more dramatic effect size. And for LIPA, it's actually among the top 1 percentile abundantly expressed gene in macrophages. So you would imagine that the cells probably develop a system to prevent it from further being upregulated somehow. We don't, although we are not sure about like exactly why, but this is what we think why we cannot further boost it. It's probably the self-feedback mechanism. And about the role of LIPA in a deeper set, I think that would be actually very interesting, just to see if we further boost up the lysosomal lipid hydrolysis in adipose, then what we'll see, whether the effects will actually be local or systematic. I think that could be a very interesting question.
Yeah, I was also wondering, are you planning to take the LIPA knockout mice also along, or is that not part of the plan?
It's not part of the plan now because Dr. Hongdu is one of our collaborator, and their group has published quite a lot for the effects of knockout. So it's completely a different cellular mechanism, right? So when you don't have any LIPA, it's simply about lysosomal lipid accumulation mess up with the lysosome function. But when express it, the effects can be not just limited to the lysosome. So we think it could be a nice comparison, but we don't really expect the opposite effects between knockout and overexpression because of that dynamics is different. Yeah.
You nicely showed this single cell RNA-seq data of the mouse plaques. I think that was mouse, right?
Yeah, that was mouse. Yeah.
And you see a subset of macrophages expressing high LIPA and one being negative. And also in the bulk RNA-seq, you see that the foam cells are the highest expressors. Is that also something you see in the single cell RNA-seq?
Yeah, for our single cell, we were not able to label foam cell at the same time. Because in the bulk, it was like they label foam cell and non-foam cell, and then they can sequence. So we were not able to do that. But the LIPA enriched cell population also has higher expression of other lysosome genes, ABCA1, FABP4. So I think this must be a population actively doing their job to metabolize lipid. So I would guess they are the foamy macrophages, although it's a guess, yeah.
And then a last question, maybe also related to the foaminess. Did you look at inflammatory gene expression along with the LIPA? Because you know also that the foamy macrophages in the plaque are not the main inflammatory ones. So how does that relate to the LIPA? How do you look at that?
Right. I'm still thinking about question. Indeed, this is something I feel intrigued about. So what has been shown is really like the LXR activation by the lipid loading, right, suppress the inflammatory responses. So this is why there's a polar response that foamy macrophage are less inflammatory. So in our case, we only observed this increased proliferation in lipid overexpression macrophage without lipid loading. Because once you give them lipid, then they begin to metabolize lipid. It's like completely different. Like none of them proliferate anymore. So I would say probably in the plaque, because of the heterogeneity, the non-foamy macrophages may be proliferating more, maybe, and maybe inflammatory and play different role from the foamy macrophage that has abundant expression of LIPA. Then when we overexpress LIPA, like give them more, then what will that lead to will be something we actually have to tackle in vivo, because in vitro will hint into the molecular mechanism, but probably not going to resemble. So our next step will be to characterize the atherosclerotic plaque size, macrophage number, perhaps also single cell RNA-seq to try to see whether it changed the subpopulation distribution.
Yeah, I'm really looking forward to these in vivo experiments. I really want to see what's happening there.
Thank you. Yeah, we'll keep you posted.
Thanks a lot again. I think we will have to conclude here. It's 5 o'clock now, at least here in Amsterdam. So thanks a lot again for sharing this new and interesting data. Next week we'll be here again, and then we will have Li-Wu Li from Virginia Tech, and he will present on innate immune memory. And actually the week after, we'll also have innate immune memory, so trained immunity. And those are the two talks that we scheduled so far. So probably in June we will stop, people are going to work again, and we will try to continue with the actual physical Immunometnet symposium, hopefully later this year, or at least in the beginning of next year. So certainly keep following us on Twitter or via the website. We will always post what we are planning to do. So meanwhile, I thank you again, Hanrui, and I look forward to actually meet you in a real meeting, and I look forward to the new data you'll be working on. Yep. So thanks a lot for your time and for your presentation. Thank you all for watching, and I'm looking forward to meet you again next week and in the future in real life.
Thank you, Jen, for making this available, for organizing it, and thanks everyone for tuning in. It's so exciting.
Thank you. Goodbye.
Bye.